Hello friends, hope you have seen our last lectures on the different types of staining procedures. In last lecture we have studied first example of the special staining, and that is cell wall staining by chances method. Now in this particular lecture we are going to observe demonstration of bacterial capsules by using Maneval's method. It is the Maneval's method. It is the procedure behind the Maneval's method to observe bacterial capsules. Notice the exact mechanism behind the demonstration of bacterial capsules. So let us see the procedure, that is chances method, for the demonstration of capsule staining. Now first of all I would like to explain what is capsule. In case of prokaryotic cell, especially in case of bacteria, algae and protozoa, they have a slimy mucilaginous viscous layer present outside the cell wall. It is present outside the cell wall. It is external to the cell wall. Suppose this is a bacterial cell. This is cell wall of bacterial cell. All of you know it is the outermost layer, that is cell wall. In some bacteria, outside the cell, there is presence of a thin, slimy, mucilaginous, viscous covering. That covering is called as capsule. So today we are going to observe this capsule. Now what is the nature of capsule? Capsules are especially made up of 98% water. It consists of large amount of water with only 2% of carbohydrates, or in some cases polypeptides are also present. So capsules are especially made up of 98% of water, and only 2% part is made up of polysaccharides, and in some cases there are presence of polypeptides. Now because of large amount of water, capsules are semi-transparent. We cannot observe capsule by simple staining. There are two reasons. One is they contain large amount of water, at the same time they are non-ionic in nature. Capsules are non-ionic, they are neutral in nature, and because of neutral nature we cannot stain them by acidic or basic stain, and that's why we require a special method. And here today we'll see the first we'll see the special staining used for capsule staining, and that is Maneval's method. Now what is the procedure behind the Maneval's method and how to demonstrate capsule by using Maneval's method. Here Maneval's method is a type of negative staining. It uses the principle of negative staining, and all of you know the principle of negative staining is. What is the principle? In case of negative staining, the part under study, suppose in this case capsule, we are going to observe capsule. So part under study do not stain, it remains colorless. At the same time other sides, that is the background or other part of the cells, are stained. In this particular case, suppose this is the cell and this one is the capsule. We are not giving color to the capsule, at the same time we are giving color to cell and outside background. So that is the principle of negative staining. We are using principle of negative staining and we will get the colorless capsule with against dark colored background and colored cell. So that is the principle. Notice the procedure behind the capsule staining. Procedure is very simple. As usual, take a slide having it should be properly washed. It should be grease free or oil free. Then take the bacterial suspension. You are given with the bacterial suspension. 24 hours old capsulated organisms are given in the suspension. You have inoculating needle. First of all, take a drop of 1% Congo red. That is the first stain of the procedure. So take 1% Congo red. Take a drop of 1% Congo red at one side of the slide. Then with the help of sterile nichrome wire loop, take the suspension and mix that suspension in the drop of this Congo red. After mixing it thoroughly, spread that mixture along the side with the edge of another slide. Suppose this is the another slide. With the edge of this slide, gently spread that mixture along the slide to form the smear. So this is the way of preparation of smear. After preparing the smear, allow to air dry. Do not heat fix the smear. So that is our first step. Now after complete air drying, when you completely dry that smear, then after complete air drying, flood the slide with second stain of the procedure, and it is Maneval stain. So apply Maneval stain on the slide after complete air drying of the smear. Apply it for 2 to 3 minutes. After 2 to 3 minutes, discard the stain. Discard the Maneval stain. You do not require washing, because we are generally avoid washing because we are not giving heat fixation. So only discard of the Maneval stain after 2 to 3 minutes, and again air dry the slide. After complete air drying, observe the slide under 100 X oil immersion objective. That is a simple 2-step procedure for Maneval's method. Now what is the observation? When you observe the slide after the Maneval's method under the microscope, this microscopic field shows the capsule. The capsules will be observed against blue background. You will get dark blue background. In that dark blue background, you will get the colorless capsule, and inner cell will be dark pink color. So you will get dark pink cell, vegetative cell, inner vegetative cell, darkly pink color. Background will be blue. So blue background, and you will get the colorless capsule. That is your observation, colorless capsule. So by this is we get we will observe the capsule by using Maneval's method. The observation is colorless capsule against dark pink colored inner cell, and background will be blue. Now after following this procedure, how we get this observation, how these capsules remain colorless, cell get pink color and background is blue, that was explained in the mechanism of capsule staining. So let us see the mechanism of capsule staining. Now this manuals explain the mechanism behind the capsule staining. According to the Maneval's method, 2 stains are used. One is 1% Congo red. The speciality of Congo red is a acidic stain. At the same time, it is a pH indicator. It is a pH indicator. How it indicates the pH? This Congo red at pH 3, it is red color. It is blue color. And at pH 6, it is red color. So in alkaline pH, in slightly acidic or alkaline pH, that is pH 6, it is red, and in acidic pH, it is blue. So it is one of the pH indicator that we have used in the first step of the procedure. Now what happens when you apply the second stain of the procedure, and it is Maneval's stain. Now what is this Maneval solution used in the second step? This Maneval solution consists of 1% acid fuchsin. Acid fuchsin is a basic stain. It is the example of basic stain. Then secondly, it consists of acetic acid, 3% acetic acid. 3% acetic acid. It also consists of 20% phenol, and 30% FeCl3 is there in the Maneval solution. So Maneval solution is prepared by adding 1% acid fuchsin, 3% acetic acid, phenol 20%, and FeCl3. When you add during the second step, when you add or flood the slide with the Maneval solution, this Maneval solution has a FeCl3. FeCl3 acts as a chemical fixative. It fixes your smear. It acts as a mordant. So FeCl3 acts as a mordant from Maneval solution. Phenol penetrate the stain. It is helpful to penetrate the stain in the capsule, because in the capsule there is our cell, and we are giving color. We want to color the cell. So penetration of stain in the stain penetration of stain in the cell was done by phenol. So phenol intensify the staining procedure. Acetic acid gives acidic pH to the manual solution. So our manual solution is acidic, and remember this property of the Congo red. In case of first acid fuchsin from the manual cell, it acts as a stain. It is properly used as a stain to stain our inner cell. Now what happens? During the first step, you are preparing the smear with the Congo red, and then when you flood the slide with the manual solution during the second step, due to the acidic property of the manual solution, your pH of Congo red become acidic and Congo red becomes blue. So your red background during the first step turns blue, and that background we will observe under the microscopic field. At the same time, this acid fuchsin in the stain is basic stain. It get attracted, it get penetrate along with the phenol, it get penetrate and it stains the acidic cell. So this basic stain gives pink color. Acid fuchsin is pink. It gives pink color to the inner cell. So you will get pink inner cell. Capsules are neutral. They do not take the color of acid fuchsin. They remain colorless. And this blue background is of Congo red. This Congo red gives blue background in presence of manual solution, because manual solution is acidic in the nature. So that is the mechanism behind the capsule staining and or Manuel's method, and by this we can easily demonstrate the capsule under the microscope. So that is about the special second example of the special staining, and it is Manuel's method for demonstration of the capsule. Now in next lecture we will going to observe third example of the special staining, and that is special staining required for demonstration of volutin granules by Albert's method. So for this time we will stop here. Stay tuned. Thank you.